Hi, friend. This is the fifth and last lecture on the digestive system. And in this lecture, we're going to cover the more of the control of the digestive system based on the neural and hormonal control. And we'll talk a little bit about disorders, not too much. So we're going to divide the gastric activity into different phases, and we'll describe the different control of each in terms of secretion. And we'll also talk a little more about the enteric nervous system and also the control of the pancreatic and hepatic secretions. And then finally, major disorders. So let's look at the regulation motility. As we saw from the previous 4 lectures, that the movement of the bolus of food is critical, as it has to move to each stage for its treatment by the different parts of the digestive system. First of all, the movement of the motility of the digestive system is divided into both skeletal and smooth muscle. We mentioned that very early on. So here I made a little table showing the control motility. So on the left side are those parts of the digestive system covered are controlled by the skeletal muscle. And on the right side, those areas of the digestive system covered by smooth muscle, controlled by smooth muscle. In the skeletal muscle, there's the oral cavity, the pharynx, the superior portion of the esophagus. Remember, the esophagus transitions from skeletal muscle to smooth muscle, and also the last portion of the large intestine. So these are more under voluntary control. Smooth muscle includes the rest of the alimentary canal. You know, it's only one thing, but it covers the whole GI tract, essentially. And this is involuntary. Okay, the control of smooth muscle motility is controlled by the autonomic nervous system, the enteric nervous system, and also pancreatic hormones. And we'll discuss a little bit about each. If you recall, the sympathetic nervous system inhibits digestive processes while the parasympathetic stimulates them. So remember, sympathetic inhibits, parasympathetic stimulates. The enteric nervous system stimulates sharp reflexes within the digestive system itself, and we also have paracrine hormones that stimulate or inhibit motility. Let's look at the regulation of acid secretion. That's another means of control of the digestive system. The parasympathetic nervous system changes gastric acid secretion during eating. Okay, and there are 3 phases of acid secretion, and they're divided into the cephalic, gastric, and intestinal phases. So in this table here, which we'll cover in detail, shows the 3 phases of secretion, acid secretion: cephalic phase, gastric phase, and intestinal phase. So C, G, I. Okay, let's look at the cephalic phase. In the cephalic phase, it's simply the sight, smell, or taste, or even thought of food will begin the cephalic phase, and it's stimulated by the vagus nerve. And it prepares stomach to receive food by increasing hydrogen ion secretion from parietal cells. So even before food enters your mouth, the cephalic phase begins. And so here in this first table, we've eliminated the rest of the table for clarity. We see the cephalic phase only requires sight, smell, taste, and thoughts of food, and by the vagus stimulation will cause an increase in hydrogen ions from parietal cells. Okay, we'll talk about the middle stuff in a little bit. Okay. During the cephalic phase, there are 4 effects. One is the direct stimulation of hydrogen ion release. Okay, so vagal nerves secrete acetylcholine that stimulates parietal cells to secrete hydrogen ions. So we see here there's a direct stimulation of the vagus nerve to the parietal cells to secrete hydrogen ion secretion. Okay, that's the simplest one. The second way is to stimulate a hormone called gastrin. Okay, and gastrin triggers hydrogen ion secretion. Okay, so gastrin is a hormone produced by the enteroendocrine cells, and if you increase gastrin, you increase hydrogen ions secretion. A third way in the action of the cephalic phase is the stimulation of histamine secretion. Okay, histamine triggers hydrogen ion secretion. So if we look back at this figure again, a second way is that these gastric enteroendocrine cells will secrete histamine, and these will affect hydrogen ion secretion from the parietal cells. The fourth way is an inhibition of somatostatin secretion. Okay, normally somatostatin inhibits acid secretion. So if it's inhibited, then there will be an increase in hydrogen ion secretion by the vagus nerve. And so here in this fourth pathway, we see that gastric enteroendocrine cells will inhibit somatostatin, which will then allow more hydrogen ion release from the parietal cell. So in the cephalic phase, there are 4 ways for hydrogen ion secretion to increase from the parietal cells: directly, through gastrin, through histamine, and through a decrease in somatostatin. Okay. The gastric phase occurs when food enters the stomach. Okay, and it's a continuum. So it's a continued stimulation from the cephalic phase, and this phase contributes to 50 to 60% of the total acid secretion. So in the gastric phase, food is entering the stomach. Okay, and we get an increase in hydrogen ion secretion, or continued increase in hydrogen ion secretion from parietal cells. And the secretion in the gastric phase is stimulated by 2 actions. First, food and distension of the stomach increases acid production through the vagus nerve and the enteric nervous system. Okay, so here the vagus nerve and enteric nervous system stimulation, okay, will proceed through the same processes that we occurred in the cephalic phase. And so these 4 can also occur in the gastric phase. A second way is that partially digested proteins stimulate enteroendocrine cells to produce gastrin. Okay, so there's an increase in gastrin, and there's an increase in acid secretion. This will increase pepsin, which will increase protein digestion, which will then again increase gastrin again. And so what kind of feedback is this? You've seen this before in SCB 203, where you have a stimulus, an increase in a stimulus, and the end result is an increase of that stimulus. If you said positive feedback, then you're correct. So here's a very classical example of positive feedback. So we see here that partially digested proteins in the gastric juice will stimulate gastric enteroendocrine cells to increase gastrin, which will then produce more partially digested proteins. And this positive feedback will also increase hydrogen ion secretion. Now, there's also a negative feedback mechanism occurs when the pH stomach reaches about pH 2. Okay. At low pH, will stimulate somatostatin secretion, and if you recall, somatostatin will decrease acid secretion. Okay, so at low pH, that will stop the positive feedback mechanism. The gastric enteroendocrine cells will increase somatostatin, and the result will decrease hydrogen ion secretion. Okay, so I've kind of included this arrow here to change the figure, because the textbook doesn't really show it that well. Now the next phase of acid secretion is the intestinal phase. And this adds another 10% of acid secretion before it is inhibited. Okay. So it is a continuum. It doesn't completely shut off automatically. So partially digested proteins trigger vagus and enteric nervous system stimulation to secrete intestinal gastrin. Intestinal gastrin stimulates hydrogen ion secretion from parietal cells. So this continues on to produce another 10% of acid secretion. Okay, so here's the intestinal phase. Okay, so this top pathway continues for a while longer. Now, at low pH and the production of lipids in the duodenum trigger the enterogastric reflex. Okay, the enterogastric reflex. The enterogastric reflex decreases vagal activity and acid secretion. So, of course, this is going to turn down acid secretion. So here we have the decline in pH and in the presence of lipids in the duodenum producing the enterogastric reflex. Okay, and this will decrease vagal activity, decrease gastric inhibitory peptide, and here there's directly will decrease hydrogen ion secretion. And I kind of got ahead of myself. In the intestinal phase, low pH also triggers production of secretin and gastric inhibitory peptide that also decrease acid secretion. Okay, so here I started mentioning here that secretin and an increase in secretin and increase in gastric inhibitory peptide (GIP) due to decreased vagus nerve activity will also decrease hydrogen ion secretion. So there's also a direct enterogastric reflex here directly, and also through the decrease in vagus nerve stimulation to increase secretin and GIP to decrease hydrogen ion secretion. So inhibition occurs at multiple levels. So here's acid secretion with all three phases back together again. Okay, so you should be able to know and describe each of the three phases. Of course, the green arrows are stimulatory and the red ones are inhibitory. Okay, so if you take each panel by itself, it's not as intimidating if you try to look at and study this all at once. So let's look at some of the digestive disorders. Okay, gastroesophageal reflux disease, or GERD, is common. It's caused by chronic regurgitation of acid chyme in the esophagus. And so the esophagus is not as protected as the stomach, and so the acidic chyme will irritate tissue. It's due to the failure of the gastroesophageal sphincter to close properly. Okay, so there's a reflux or backup through the sphincter. It can be due to a number of reasons, such as obesity, caffeine, a hiatal hernia infected by a bacteria called Helicobacter pylori, which produces gastric ulcers. So gastric ulcers are due to a bacterial, infection, and of course it's treated with antacids, histamine blockers, and proton pump inhibitors. Okay, so antacids act as buffers. Histamine blockers will prevent histamine from causing acid secretion, and proton pump inhibitors will prevent hydrogen ions being pumped out to produce HCl. Emesis, well, this is just vomiting. Okay, and it can be due to relaxation of smooth muscle of the stomach and small intestine, and the relaxation of esophageal sphincters. Okay. And normally the uvula closes off the nasopharynx, but if you ever threw up orange juice, it's not perfect, and you can feel your nasal cavity burn because it's not protected as well from the acid of the stomach. And of course, the epiglottis covers the glottis to prevent any of it to enter your respiratory system. Okay, now let's go back to controlled secretions. Okay, let's look at bile secretions. Remember, bile is produced by the liver, stored in the gallbladder, and released by the gallbladder through its contractions. Okay, So bile secretions is controlled by a hormone called cholecystokinin, or CCK, from duodenal cells, and these contract the gallbladder to cause bile secretion. Secretin also stimulates bile production and release. There's also a positive feedback as bile is returned to the liver from the hepatic portal vein from the ileum, which will cause more to be released. Okay, so let's look at this figure here. Duodenal cells secrete cholecystokinin, CCK, and secretin into the blood. So here are the duodenal cells here. Okay, they're secreting secretin and they're secreting cholecystokinin into the bloodstream. Now, cholecystokinin triggers contraction of the gallbladder, okay, here, which releases bile, and bile enters the duodenum of the small intestine. Okay. Now, the bile salts and secretin trigger the release of more bile. An example of positive feedback. So, as secretin and bile salts enter back to the liver, to the hepatic portal vein, it causes the release of more bile. So bile secretion is an example of positive feedback. How about pancreatic secretions? Pancreatic secretion increases during eating from parasympathetic and hormonal stimulation. Okay. Cholecystokinin, which we just talked about, secreted in response to lipids and partially digested proteins. Okay. Acinar cells stimulate the secretion of enzymes and other proteins. Okay, so in this figure here, we see duodenal cells release secretin as cholecystokinin into the blood. The cholecystokinin binds the receptors on acinar cells, the pancreas, and secretin binds duct cells. And the result, this binding triggers secretin secretion of enzymes and bicarbonate ions from the pancreas into the duodenum. So CCK and secretin also has effects on secretion of the pancreas in addition to bile. And secretin in response to acid and lipids by the duodenum, or in response to acid lipids in the duodenum, and increases secretion of bicarbonate. Okay. So here's secretin, and it causes release of bicarbonate, which is part of the pancreatic juice secreted into the duodenum. And so here is a summary of a secretion of motility of all the things we covered so far. Okay, so here's a little handy dandy table. Okay, finally, let's talk about the final stages of the digestive system. So things that are not digested or absorbed will have to be eliminated through defecation. So let's talk about the defecation reflex. Okay, this is due to mass movements which force fecal material into the rectum. Okay, and the defecation reflex is due to stretch receptors that transmits the sensation of rectal distension to the spinal cord. So it's the movement of the fecal material that will stretch receptors that provides the sensation for the defecation reflex. Okay, so let's look at this process that occurs in the defecation reflex. Okay, so of course, this part's an involuntary reflex. Stretch receptors transmit the sensation of rectal distension to the spinal cord. Okay, so here's the distension from the fecal matter entering the rectum that sends stimulus to the spinal cord. Parasympathetic neurons cause smooth muscles in the sigmoid colon and rectum to contract. Okay, so that will cause movement, forcing the fecal material from the rectum and the sigmoid colon, and also relaxation of the internal anal sphincter, which is under involuntary control, to relax, to allow it to move. Okay, so here we see parasympathetic neurons cause smooth muscles in the sigmoid colon and rectum to contract and the internal anal sphincter to relax. Finally, the cerebral cortex triggers relaxation of the external anal sphincter, because the external anal sphincter is under voluntary control. And contraction of the anal muscles allow feces to pass out of the body. Okay. So even though that the internal anal sphincters are relaxing, as adults, once you gain control of the voluntary control, you control when you want to continue the defecation reflex or not. Okay, so here is the last step. Impulses from the cerebral cortex trigger relaxation of the external anal sphincter. The external anal sphincter and contraction of the muscles, allowing feces to pass out of the body. Okay, so it requires both. The reflex requires both the involuntary control as well as voluntary control. Okay, so finally, let's talk a little bit about the effects from changes in the large intestinal motility. Okay, because you could have a hyperactive large intestine, which causes things to move faster, or you could have very low motility where things move slower, and this can have very profound effects on defecation. So motility changes of the large intestine affects water present in feces. Okay, when motility increases, watery feces or diarrhea occurs because there's less time for water to be absorbed. Okay, I think you can understand that. If motility decreases, too much water is absorbed, and you end up with a harder feces, and constipation occurs. The harder feces does not move as easily as does a more watery feces. Okay, so the effect on motility can have an effect on the amount of water present in the feces. So the final figure is just the review of the digestive system that includes everything from the beginning to the end. Okay, so this is a nice handy dandy table to review the major steps that occur throughout the digestive system. And this was a very important topic as we spent 5 lectures on it. Okay. And hopefully they were broken up into 5 smaller lectures rather than several smaller, larger lectures. So this concludes the digestive system. Okay. Next, we're gonna our next lectures will go on to a related topic called of nutrition. Because so far we didn't talk about the types of food you enter in your body. Next, we're going to talk about the different types and quality.